Welcome to the lecture on kidney autoregulation. Part 2. My name is Olga Sosnovtseva, Department of Biomedical Sciences, University of Copenhagen. One of the most striking characteristics of the renal circulation is the ability of the kidney to maintain a constant renal blood flow and glomerular filtration rate as renal perfusion pressure is changed. Such regulation is mediated by two mechanisms: tubuloglomerular feedback and renal myogenic response. Tubuloglomerular feedback involves a flow-dependent signal that is sensed at the macula densa and changes radius of the afferent arteriola. The myogenic response involves a direct vasoconstriction of the afferent arteriola when transmural pressure is increased. The current view is that these mechanisms act together, and their primary role is to stabilize renal function by preventing pressure-induced fluctuations in the renal blood flow and glomerular filtration rate. To model nephron autoregulation, we should take into account these two regulatory mechanisms. The essential elements and mechanisms of the model are summarized in the causal loop diagram in this figure. Notice, please, that signs at the arrowhead indicate the direction of change of a given variable in response to a change in the variable at the arrow's tail. The elements of the tubuloglomerular feedback loop are shown in this figure to the right. The loop includes glomerular filtration, the mass transport of sodium chloride and water, the reabsorption of sodium chloride and water in various tubular segments of the loop of Henle. Reabsorption of sodium chloride is dependent on flow rate in several of these segments. Therefore, the concentration of sodium chloride in tubular fluid at the macula densa is strongly dependent on the flow rate of tubular fluid. The signal from the macula densa produced in response to variations of sodium chloride concentration in tubular fluid propagates and changes the vascular resistance of the afferent arteriola. The change in vascular resistance affects glomerular filtration rate, closing the loop and providing negative feedback. The myogenic mechanism is represented by the loops in this figure to the left. The period of oscillations is determined by instability in membrane potential caused by an interaction between calcium and potassium currents. The magnitude of oscillations in vascular diameter is determined by the level of intracellular calcium. These mechanisms act together. Tubuloglomerular feedback affects the membrane calcium conductance, allowing tubuloglomerular feedback to modulate both the amplitude and frequency of the myogenic mechanism. For simplicity, we can remove detailed description of myogenic response, focusing on tubuloglomerular feedback regulation. This simplified causal loop diagram illustrates the main elements of tubuloglomerular feedback mechanism. If the rate of glomerular filtration due to, for instance, an elevated arterial blood pressure is too high, the active reabsorption of sodium chloride in the loop of Henle is not complete, and the salt concentration at the macula densa is too high. The macula densa cells then produce a signal that causes constriction of afferent arteriola and reduces glomerular filtration rate. This feedback is negative. The feedback involves also a delay associated with the time it takes the filtrate to flow through the loop of Henle and to transmit the signal. Flow diagram helps to put together all elements of the single nephron model. There are 5 main processes implemented into the model. The blood flows from the arterial system through the afferent arteriola, the glomerular capillaries, and the efferent arteriola to the venous system. The blood flow through the glomerulus capillaries is determined by the pressure difference between the arterial and venous sides and by the two flow resistances. The glomerular filtration rate depends on the difference between the glomerular pressure and the proximal tubular pressure, accounting of course for the difference in protein osmotic pressure across the capillary wall. Flow through the loop of Henle, where reabsorption of sodium chloride and water takes place. Macula densa cells monitor sodium chloride concentration and send a signal to afferent arteriola to change its radius. Constriction of the afferent arteriolar radius causes a reduction in blood flow into the glomerulus. The first equation of the model represents the pressure variations in the proximal tubula in terms of in- and outgoing fluid flows. It takes into account three processes: glomerular filtration, flow in the loop of Henle, and reabsorption in the proximal tubula. Arteriolar response is modeled by a second-order differential equation in the form of damped oscillator. Note that it can be rewritten in the form of two first-order differential equations. Delay in the system is modeled as a third-order lag equation. The expressions for functions used in the model involve a number of algebraic equations that must be solved along with integration. These expressions were obtained by approximation of available experimental data. Let us see how the model can reproduce experimental observations. This figure shows experimental recordings and computer simulations of proximal tubular pressure. It is clearly seen that the model reproduces oscillations of the same frequency, amplitude, and level of noise. Let me remind that renal autoregulation is important to maintain a stable renal blood flow and glomerular filtration rate despite changes in systemic blood pressure. Let us see how dynamics of the model will change with increasing blood pressure. The first figure shows how the radius of afferent arteriola changes with increasing blood pressure. One can see that there is a range of pressure where the vessel constricts. The second figure shows that the renal blood flow remains constant within a wide range of arterial pressure. Let us see now how properties of tubuloglomerular feedback change dynamics of the model. For small values of parameter defining efficiency of tubuloglomerular feedback mechanism, we observe periodic oscillations in the proximal tubular pressure, similar to oscillations observed experimentally for non-tensive rats. With increasing parameter, irregular oscillations occur that are typical for hypertensive rats. Let us check now whether the model can simulate interaction between neighboring nephrons. Let us start by considering the vascular coupling. The muscular activation arises and travels backwards along the afferent arteriola in a damped fashion. When it reaches the branching point with the arteriola from the neighboring nephron, it may propagate in the forward direction along that arteriola and start to contribute to its vascular response. In our model, this type of interaction is represented by adding a contribution of the muscular activation of one nephron to the activation of the other. The vascular signals propagate very fast as compared with the length of the vessels relative to the period of tubuloglomerular feedback oscillations. The vascular coupling can therefore be considered as instantaneous. Such interaction produces in-phase synchronization between tubular pressure oscillations in neighboring nephrons. A hemodynamic interaction occurs when one nephron is stimulated by tubuloglomerular feedback mechanism to contract its afferent arteriola. This causes the hydrodynamic pressure to rise at the upstream branch node, increasing the blood flow to the second nephron. Half a period later, when the increased blood flow activates the tubuloglomerular feedback mechanism in the second nephron and causes its efferent arteriola to contract, the blood flow to this nephron is reduced, and the blood flow to the first nephron increases. Due to the time lag in the response, this type of interaction tends to induce antiphase synchronization between nephrons. Nephrons are arranged in a complex branching structure that delivers blood to each nephron and at the same time provides a basis for an interaction between nephrons. The renal vascular network has a complex topology specific to the kidney. To implement a model of multiple nephrons interacting through vascular network, one will need a single nephron model and a vascular model. They should be combined together, taking into account conservation of blood flow at each branching point, vascular propagated signals via activation of smooth muscle cells, hemodynamic interaction via blood redistribution, probability-based topology of vascular network. This is a challenging task, both from physiological and computational points of view. Thank you.